Welcome to Module 12, which is an introduction to immunology and serology. Serology is testing a patient sample for the presence or absence of specific antigens and/or antibodies. Testing in the serology department revolves around the detection of antigens or antibodies in a patient's sample. We call this in vitro testing in the test tube because that's where the reaction will occur and become visible to the naked eye. To understand serology testing, you must have a concept of antigens and antibodies. These are crucial for the principle of the test and how these are created. An antigen is any substance which is recognized as foreign. It's non-self, it's not you, by the body, which will result in an immune response. In serology, it's usually the disease-causing agent, the pathogen, which we're looking for. A pathogen is an agent which causes infection or disease, especially microorganisms such as bacteria, viruses, or parasites. An antibody is a blood protein which is produced, which is specific for the antigen which caused its production. These are proteins that bind to the offending antigen, and once they bind, it's recognized as foreign by the body, and it results in its destruction. The primary function of antibodies is to protect us from disease. Digging a little bit deeper into the immune response, there are two types of immune responses in your body, the humoral immune response and cell-mediated immune response. Serology focuses on the humoral immune response, which is the antibody production to pathogens that are invading your body. The humoral immune response involves lymphocytes, a type of white blood cell. These are the only cells in the human body which can produce antibodies. There are many types of lymphocytes. The B lymphocyte is the specific type of lymphocyte which produce antibodies. The initial immune response to a pathogen takes days or weeks to become effective. Once a B lymphocyte is programmed to produce these specific antibodies, they have lifelong memories. We have this immortal line of cells that will always remember this antigen, and if it invades the body again, the response is immediate, and there's a spike in the antibody production and a rapid destruction of the pathogen before it can set up an infection in your body. In the immune response, there are two ways the humoral system is activated: by vaccination, which is a passive immunization, and getting it through disease, which is an active immunization. The most important method of preventing disease transmission is through vaccination. What they do is they take a piece of the offending microbe, not the whole organism, just the piece that is most foreign to the human body. For example, the rubella virus. And then they take this piece and they inject it into your arm. Your body then recognizes this piece, which is common to all rubella viruses, as foreign and mounts an immune response, resulting in the formation of antibodies specific for the rubella virus. If the individual is exposed to the virus, their immune system will immediately start producing antibodies because I've seen this antigen before, I didn't like it then, I don't like it now, I know how to do this job. So massive amounts of antibodies are produced that will then result in the destruction of the specific virus or organism. Prior to vaccination, a rite of passage through childhood was actually being infected with the pathogen. Chickenpox was a disease nearly everyone got during childhood. The virus enters the body, illness occurs, the immune system is activated, but you had to suffer through that miserable illness. Most of you will never know that suffering because you have been vaccinated, but these little red bumps that became pustules that itched horribly would form. Immune memory cells are produced during the disease process, which, if the virus enters your body again, will immediately go into action to produce antibodies which will immediately cause destruction of the virus. Back in the old days, mothers would take their children to houses where these infectious diseases, mumps, measles, chickenpox, so that their children would get over it quickly. They wouldn't have to wait for some random event. Now, keep in mind, getting the disease is not without risk. For example, measles, especially if it's transmitted to a pregnant woman, can cause birth defects in her developing fetus and even death of the fetus. Children who are sick with these childhood diseases, it puts their immune system down to where they're prone to get other infections which may kill them. So vaccination has really decreased the mortality rate in children due to these infectious agents. We cannot see antigens and we cannot see antibodies. Serological tests have been developed to detect the endpoint where an antigen actually attaches to an antibody, resulting in a visible or measurable product. Knowing the principle of each type of testing methodology is crucial to the practice of serology, and this will be what will keep you up at night when studying all the different methods performed for serological testing. When I'm teaching serology, the principle of the test is so crucially important. And the principle is what is reacting with what to give a positive reaction. Sometimes we're looking for the actual antigen in a patient sample. Sometimes we are looking for specific antibodies in a patient sample. Antigen tests are ideal as it can catch infections early on and allow specific treatment if they are available. Antibody tests are not as reliable as it may take days or weeks for them to become detectable in the body. So this is what we call a false negative, in which you've been infected with the microorganism, but a clinically significant amount has not been produced yet to give a positive result. So this is when you go to the doctor's office and they do these little kit tests on you. There can be false positives or false negatives. It's your job as an MLT to read that product insert to know what can cause false positives and false negatives in a patient sample, and there are many. We're going to start talking about the serological techniques. I want you to know that in any serological test, one of the reactants will be known and one reactant will be unknown, which is the patient sample. Sometimes the antigen is known and sometimes the antibody is known. It will depend upon the reactants provided in the testing system. So if we're using a known antibody, then what we're looking for is an unknown antigen in the patient sample. If we are working with a known antigen, so just like COVID-19, then we're looking for the antibody to COVID-19 in the patient sample. So again, in all test systems, there will be one known, and then the unknown is found in the patient sample. I want you to know the difference between a qualitative test and a quantitative test. Most kit tests are qualitative. The result is either positive or negative. We don't know how much of the stuff we're looking for is present, but we just know that it's there. Most kit tests give this type of result. For example, a flu test. Some tests are quantitative, and these are performed on a machine. And the reactant we are looking for is not only present, but the exact quantity is measured. For example, this is crucial for HIV viral loads to determine that therapy is working. So a patient's diagnosed with HIV, they do a viral load, and it's very, very high. So they put the patient on therapy, and there's many different therapies available, and the patient then is tested a certain time period after the therapy starts. If the therapy is working, the viral load will decrease. If it's not working, it will remain the same or go higher. So this is used to modify the treatment of the patient. On this slide, there is a list of techniques which you will become intimately familiar with during the serology course. We're going to discuss each one on the following slides. Each of these techniques have many different applications, and they're used to detect either antigen or antibody. The precipitation technique involves having two soluble reactants, antigens and antibodies, which interact and form an insoluble product. One is known, which is the reagent, and one is unknown. So if we're looking for antigen, we're going to have a known antibody. If we're looking for antibody, we're going to have a known antigen. And so again, these are in liquid. You mix them together and you cannot see them, but if whatever reactant is present that we're looking for is present, then these particles form, which eventually get so hard they drop to the bottom. It's kind of like making rock candy, which many of us did as children. So you take water and supersaturate it with sugar, meaning you dissolve more into it as much as you can until crystals fall out by themselves. Then you place a string in the water. The sugar will recrystallize on the string. It's reacting with components in the string. And imagine the string is the antigen and the sugar molecules are the antibodies. And I've listed a link just for fun if you would like to do your own experiment and make your own rock candy. So this picture illustrates the initial solution, which has soluble antigens and antibodies. And again, if the substance we're looking for is present, that precipitate will form, and that would be a positive reaction. A very simple definition of the next technique called agglutination is the clumping of particles. In serology, the particles are usually either red blood cells or latex particles which have been coated with something. So the principle, the particles contain the antigens. The antigens are mixed with a fluid which contains the antibodies. And if clumping occurs, the test is positive. The most common application is in immunohematology, also known as blood bank. So this shows the red cells being tested with two types of antibodies, anti-B and anti-A. So the top picture, you have the type A with anti-B. Because these red cells do not have the B antigen, there's no binding. They're free in solution around the red cells. In picture B, where there's agglutination, you have a type A blood cell, an anti-A antibody, that's the reagent, and as you can see, binding does occur. So you see there's a binding of this antibody with antigens on one cell, binding of this antibody with antigens on another red cell. When you get enough of this going on, then if you look at the picture, you can see the clumping of cells on the left, which is a positive reaction for agglutination. If the antigen-antibody reaction does not occur, you have this smooth, silky appearance to the red cells, which is illustrated on the right. I want to emphasize again that in serology, we have these invisible antigens and antibodies. One is always going to be known, one is unknown. And what we want to do is make them say, hi. I want to see a visible reaction where I know that the reactant I'm looking for is present. Or if there is no reaction, that the thing I'm looking for is not present. In immunoassays, there's three different types of reactions I want you to be familiar with. The first is immunofluorescent. If an antigen and antibody bind, the sample fluoresces. So what we're going to do is we're going to tag the known with a fluorescent probe, and the unknown will bind. And then we wash the slide away. And if this binding occurs, it sticks, and we look at that under a fluorescent microscope. And as you can see in the picture illustrated on the top, we can see a fluorescent reaction occurring. If there is no fluorescence, then it's negative. The second type, which is one of the most commonly utilized techniques, is the enzyme-linked immunosorbent assay. We also call it ELISA. If antigen and antibody bind, it results in a colored line forming. So look at the bottom picture. We have S, which is the sample well, where you apply the patient's sample. Then T is the test, C is the control. So for the test line, if the substance we're looking for is present, it will bind at this line. and then after a certain period of time, a color will form, which is a positive reaction. If no color forms, it's a negative. Well, how do we know everything's working properly? Well, we have C, which is the control line. The control line must always be positive. It gives you, number one, an example of what a positive looks like, and number two, it lets you know that the reagents in the system are working properly. Radioimmunoassay, RIA, the patient's sample is added to particles with a radioactive tag. If the patient's sample contains the substance, antigen or antibody that we're looking for, it will attach to the particles. Then a wash is done and the radioactivity is measured. So if you have a high level of radioactivity, that's a positive, and no radioactivity is a negative. And this most frequently is done in a quantitative method because the amount of radioactivity present is directly proportional to the substance present in the patient's sample. We're going to talk about two techniques used in immunochemistry. The first is chemiluminescence. This is an antigen-antibody reaction detected by the emission of light, luminescence. Glow sticks are the perfect example. When you snap the stick, the chemicals mix and emit light. Another example of this reaction is luminol, which is frequently used in forensics to determine biological stains are present, primarily blood. So you see them spray this solution and then you use a special light and fluorescence will occur. This gives a quantitative result when done, and it takes a special piece of equipment to do this. Fluorometry, which is also known as fluorescence spectroscopy, detects the presence of antigen-antibody reactions by fluorescence. The amount of fluorescence present, if any, is measured, and this gives a quantitative result. Molecular diagnostics is a rapidly growing field. Eventually, this will be a separate course in all laboratory science curriculums as the list of tests and application grows at an incredibly rapid pace. Molecular diagnostics has many different applications, including the diagnosis of genetic diseases, cancer, and infectious diseases. In serology, we will focus on the diagnosis and monitoring of infectious diseases by the detection of genetic markers in the DNA or RNA of the target. An integral part of learning about this field will be in the understanding of the polymerase chain reaction, PCR, which revolutionized this field. The polymerase chain reaction, called PCR, is like a Xerox machine for DNA. It is used to make many copies, millions or billions, of a particular region of DNA. This is a very simplified explanation of a technically complex procedure. The target region of DNA is a specific size of nucleotides. After PCR, the components in the solution, after replication, are separated by electrophoresis. Electrophoresis is a process performed in agar gel where the solution is added at one side, an electrical current is applied, and the components in the solution are separated out by size. The little bitty ones, they get really sparked by that electricity, and they move very fast down the gel. The big ones, you just kind of keep poking them, and they move much more slowly, again, because of their size. The history of PCR is fascinating. Take some time to visit this link. I'd like to give a better description of the principle of PCR. So it's a technique to make many copies of a particular section of DNA. So we add the patient's DNA to a little tube, and then we add a reagent that will snip it so that we have the sizes, the target of what we want to replicate. Then we add a chemical that separates the DNA into two separate strands. Once these are separated, then nucleotides are present that will rebuild the missing strand. And this is done through a regulation of temperature. It's put into a thermocycler, and the thermocycler heats, which causes the DNA to separate, and then cooling, which allows these other nucleotides to rebuild those branches of DNA. So now we have twice as many as we did before. And you program the thermocycler depending upon what you want and how long it takes for usually 20, 40 cycles. Once the thermocycler has completed its job, then the sample is placed into agarose gel. And they will then migrate according to the size. As I said before, the large particles have a really hard time moving through the matrix of the agarose. It's relatively thick, whereas the really small fragments, boom, they just zip on down to the bottom. So we run a known to where we know what the different fragment types are. And then that is compared to our results, and we can then identify whether or not the target that we're looking for is present. Really cool procedure. As time goes on, molecular diagnostics methodologies utilized are becoming more simplified and automated. And anytime it goes into a black box of automation, then it can be easily performed by a variety of different people in different venues. A busy obstetrics gynecology office can now purchase a kit, which are run by a machine that will detect chlamydia, trichomonas, and gonorrhea from a single sample in a matter of hours, whereas it used to be sent to a lab and different tests had to be done, and it could take a long time. Clinical hospital laboratories can screen patients and employees for methicillin-resistant Staphylococcus aureus, which is a real problem in infections acquired during hospital stays. So they screen the patients and the employees, and if they test positive, they're isolated and given medication to get rid of the infection. Most of the tests, though, are very infrequently ordered, and these are extremely expensive to run on site. From the equipment that's needed to do the testing, to the actual kits themselves. The reagents for the HIV test can cost $1,200 or more per kit, and the kit can test 120 samples. And you don't want to be the individual responsible for a failed run. And then there's $1,200 plus all the other time for reagents and supplies and people involved. This is just for the kit and does not include the cost of consumables, your pipette tips, your buffers, your other chemicals. It also does not include the cost of the automation to run it, which is in the hundreds of thousands of dollars. Reference labs perform the bulk of molecular testing as they have enough volume to turn a profit. But again, for certain tests, for certain molecular tests, they're now being able to bring down the cost to where you can run a limited menu in your laboratory. The most common type of serological tests performed are the kit tests. And these usually utilize the ELISA techniques, where you have the line is positive, it appears it's positive, if it doesn't appear it's negative. Although the methodologies used are very simple, the principle, what reacts with what to give a positive reaction, are crucial in performing the tests. The kits have built-in positive and negative controls, which must be interpreted without error. I was laboratory director for physician office labs, and these people just don't get it because they weren't trained to be a medical laboratory technician. And it took a lot to get them on track with forms that made them interpret everything on the test. The steps must be followed exactly. The correct number of drops, the correct number of swirls if that's involved in the procedure, and the timing of each step. In fact, the manufacturer will state if results are read that exceed the time limit, the results are invalid. And again, I just had an incredibly hard time convincing these folks that you can't just run off and leave it. You have to have a timer, and if it exceeds the timer, that you're off doing something else, then you have to repeat the test. Timing is crucial because false positives or negatives may occur if the timing is off. Some examples are flu, strep, pregnancy, infectious mono, and COVID-19. The serological kit test may be spread out by department. For example, the strep testing is done in microbiology. D-dimer, which is the type of coagulation test, is done in the coagulation department. ABO blood typing is done in immunohematology. Fluorescent and RIA procedures are performed in special chemistry. So this is why we don't have a specific rotation for serology, because they're not done in just one department. They're done in a variety of different departments. So, in summary, serology is based on the detection of immune substances, either antigens or antibodies, in a patient sample. A variety of techniques have been developed to detect immune substances to assist in the diagnosis, monitoring, and treatment of diseases. Knowing and understanding the principle behind every testing methodology is crucial to the practice of serology. If false positives or negatives occur, you will have an understanding of why. Most laboratories, again, will not have a serology department. Testing is performed in the department in which the results are the most significant. Although many tests are easy to perform, all processes such as quality control, which we must run samples that are known positives and known negatives to validate the kit test or other tests before use. The correct interpretation, because sometimes a positive visible reaction is really a negative reaction. It can get very confusing. And again, these processes must be followed at all times. So this concludes the unit of instruction, and you can now go take the quiz.